January 20th, 2013. Lake Whillans, Antarctica. 2 miles beneath a team of American scientists, a superheated jet of sterilized water melted through the last inches of ancient ice, piercing a liquid world that had been sealed in darkness for millions of years. This was a mission the world's leading polar experts had called impossible. Their rivals, the legendary Soviet researchers, had declared this realm a frozen tomb, scoffing at the idea that anything could survive here. But the samples pulled from that dark water would provide the stunning conclusion to the story of how Soviet scientists mocked U.S. researchers in Antarctica. Then they found the oldest life on Earth. A Cold War of ideas. For much of the 20th century, Antarctica was more than just a scientific frontier. It was a theater for the Cold War, a place where the United States and the Soviet Union vied for dominance not with weapons, but with drills, tractors, and scientific prestige. And at the heart of this rivalry was a fundamental disagreement about what lay hidden beneath its colossal ice sheet, a sheet of frozen water so immense that in some places it is over 3 miles thick. This monolithic icy desert, larger than the United States and Mexico combined, seemed to be the very definition of a lifeless wasteland. The Soviet Union had long been the pioneers of Antarctic exploration, planting their flag in the harshest corners of the continent. Their Vostok station, established as part of the International Geophysical Year in 1957, was a testament to their ambition and endurance. Perched atop the high polar plateau, nearly 11,500 feet above sea level, it is one of the most remote human outposts on the planet. It holds the record for the coldest temperature ever recorded on Earth, a mind-numbing minus 128.6 degrees Fahrenheit, a temperature at which steel can shatter like glass, and human breath freezes into crystals before it hits the ground. Vostok was a symbol of Soviet resilience and scientific might. It was here, using seismic soundings that sent shockwaves through the ice to map the bedrock below, that a young Russian geographer named Andrei Kapitsa first theorized the existence of a vast, liquid lake trapped far beneath the station in the late 1950s. For years, it remained a theory, an anomaly in the data. But by the 1990s, international remote sensing technology, including airborne ice-penetrating radar, confirmed his hypothesis in stunning detail. Lake Vostok is one of the largest lakes in the world, with a surface area of nearly 6,000 square miles, all buried under 2.3 miles of solid ancient ice. Geologists estimated it had been utterly sealed off from the outside world, from the sun, from the atmosphere, for an estimated 15 to 34 million years. It was a geological time capsule on an unprecedented scale. From the moment of its confirmation, the Soviet and later Russian scientific establishment viewed Lake Vostok through a specific and very practical lens. It was a perfect, sterile sample of a bygone world. Their primary interest was in paleoclimatology, the study of past climates. By drilling deep into the ice sheet above the lake, they could extract ice cores, long cylinders of ice that contain trapped air bubbles from ancient atmospheres. Each layer of ice is like a page in Earth's diary, giving them a pristine record of temperature, atmospheric composition, and volcanic events going back hundreds of thousands of years. For this type of work, a little bit of surface contamination was an acceptable nuisance. The idea that this isolated, dark, high-pressure environment could harbor life was, to them, preposterous. It defied all known logic of biology. Early Soviet analyses of ice cores taken from just above the lake surface, ice known as accretion ice, which is formed from the lake water freezing to the bottom of the glacier, reported no viable cells. Any faint biological traces, any stray fragments of DNA, were immediately and emphatically dismissed as contamination from the drilling process itself. The Soviets used a drilling method that was brutally effective but biologically messy. To keep their deep borehole from freezing shut under the immense pressure, they filled it with a mixture of kerosene and freon. This anti-freeze cocktail worked, but it meant that anything brought up from the depths was bathed in a chemical soup teeming with surface microbes. To them, any bacterium found was simply a hitchhiker, a stray from the surface world carried down with the drill. This sterile void theory became scientific dogma. The prevailing Soviet view, echoed by many cautious scientists in the international community, was that the combination of factors made life an absolute impossibility. First, there was the total and eternal darkness. With no sunlight penetrating the miles of ice, photosynthesis, the foundational process for over 99% of life on Earth, was out of the question. Second was the extreme cold. The water was believed to be around 27 degrees Fahrenheit, kept liquid only by the immense pressure of the ice above and the faint geothermal heat from below. Third, there was the crushing pressure, estimated to be over 350 times that of our atmosphere at sea level. Finally, and most critically, there was the perceived starvation. Sealed off for millions of years, what could anything possibly eat? The lake was thought to be ultra-oligotrophic, a scientific term for being incredibly nutrient-barren. The skepticism in the international community was palpable, and the Soviet researchers, rightfully proud of their Antarctic dominance, did little to discourage it. They were the masters of the ice, the ones who had endured the cold and done the hard work. In conference halls and informal conversations, they would often privately and publicly mock the fanciful and unscientific ideas coming from some American scientists who suggested otherwise. But across the globe, in the labs of American universities, a different idea was taking root. A small but persistent group of U.S. researchers, led by figures like microbiologist John Priscu from Montana State University, looked at the exact same set of hostile conditions and saw not a tomb, but an opportunity. They were part of a new generation of scientists who studied extremophiles, organisms that thrive in conditions that would be instantly fatal to humans. The discovery of vibrant ecosystems around hydrothermal vents on the deep ocean floor in the 1970s had already shattered the paradigm that all life depended on the sun. These organisms were chemosynthetic. They ate chemicals like sulfur and iron billowing from the vents to create energy. Priscu and his colleagues argued that a similar process could be happening in Lake Vostok. They theorized that faint geothermal heat from the Earth's core could warm the lakebed and that minerals leaching from the rock could provide the chemical food source for a unique, self-sustaining ecosystem. They pointed to tantalizing hints found in that very same accretion ice the Russians were drilling. In the early 2000s, U.S. analysis of this ice, which the Russians generously provided through international collaborations, revealed microbial DNA fragments. Though the Soviets dismissed these as more contamination from their dirty drill fluid, for the Americans, it was the first clue, the scent of a biological mystery. For them, it wasn't a question of if life could exist there, but how. The stage was set for a direct challenge to the old guard, a scientific showdown on the ice. The Cold War of military and political ideology was over, but a new, intellectual Cold War of scientific philosophy was just beginning. But proving it meant drilling where no one had ever drilled before, and doing it in a way no one had ever imagined, a mission against all odds. Challenging the scientific consensus established by the world's leading polar experts was no small task. The American scientists, galvanized by the possibility of a hidden biosphere, knew they couldn't just present a theory. They needed irrefutable proof. The legacy of the Soviet drilling at Lake Vostok cast a long shadow, a cautionary tale of biological ambiguity. Any biological sample they collected would be immediately scrutinized by a skeptical world, and if there was even the slightest chance of contamination, their findings would be dismissed, just as the earlier DNA fragments had been. The burden of proof was on them, and it was immense. If they were going to prove life existed under the ice, they had to do something the Soviets had never managed: drill with almost perfect, unimpeachable sterility. The story of how they achieved this is a masterclass in ingenuity and ambition, a testament to overcoming monumental engineering hurdles. Thus was born the WISARD Project, an acronym for Whillans Ice Stream Subglacial Access Research Drilling. It was a massive $10 million undertaking funded by the National Science Foundation, bringing together a consortium of top American universities and scientists. This wasn't a small-scale experiment. It was a polar exploration mission on the scale of a NASA project, involving dozens of scientists, engineers, drillers, and support staff. Their target was not the immense, and deeply buried Lake Vostok, a challenge too great for a first attempt, but a smaller, shallower body of water named Lake Whillans. This lake, situated under the fast-moving Whillans ice stream in West Antarctica, was still buried under more than 2,600 feet of ice, but its relative accessibility made it the perfect test case. What made it even more compelling was its location. It was part of a dynamic network of subglacial rivers and lakes that scientists now realized acted as a plumbing system lubricating the base of the ice sheet. Finding life here wouldn't just be a biological curiosity. It could have profound implications for understanding how Antarctica's ice moves and melts. More important than the location was the method. The Whissard team knew they couldn't use kerosene and freon. Their mission was fundamentally biological, and their methods had to reflect that. Instead, they designed and built a revolutionary hot water drill, a beast of engineering specifically tailored for clean access. The system was essentially a giant, high-tech pressure washer designed to operate in sub-zero temperatures. It would use powerful heaters to bring water to nearly 200 degrees Fahrenheit and then blast it through a high-pressure hose down a borehole, melting its way through the ice sheet like a hot knife through butter. But the real genius was not in the heat, but in the hygiene. The filtration system was the heart of the entire operation. All the water used for drilling, drawn from melted surface snow, was first passed through a series of filters that could remove 99.9% of all particles, down to the size of the smallest bacteria. After filtration, the water was blasted with a powerful dose of germicidal ultraviolet light, a process designed to destroy the DNA of any remaining microbes, rendering them inert. The result was torrents of water cleaner than anything found in a surgical theater. The entire system, from the hoses to the drill head, was designed to ensure that the number of bacteria introduced into the lake would be less than 1 in 10,000 of the bacteria they expected to find. They were building a machine to find life, and they had to be absolutely certain they didn't bring it with them. This commitment to cleanliness was their answer to the decades of Soviet skepticism. The logistical challenge was staggering, a campaign of its own. The entire drill rig, a collection of massive custom-built shipping containers housing the heaters, powerful pumps, filtration systems, and mobile laboratories, had to be hauled over 1,000 miles across the treacherous Antarctic landscape on enormous sleds pulled by giant tractors. This convoy, known as the South Pole Traverse, moved at a walking pace, navigating fields of hidden crevasses and brutal weather. The team of 50 scientists, engineers, and support staff would be working for months in one of the most hostile environments on the planet, with temperatures regularly dropping to minus 30 degrees Fahrenheit and constant, soul-crushing winds. The skepticism was not just from their Soviet rivals. Even within the Western scientific community, some thought the project was an overly ambitious, high-risk, and expensive gamble. The pressure on co-chief scientist John Priscu and Brent Christner was immense. They had made a bold claim that challenged a scientific superpower. They had convinced funding agencies to invest millions of dollars. Now, after years of planning, building, and a grueling journey across the ice, the world was watching to see if they could back it up. In January of 2013, the Whissard team was finally in position. The massive containers were arranged, the drill tower was erected, and the complex web of hoses and cables was connected. The silence of the Antarctic plateau was broken by the roar of the generators. The moment of truth had arrived. They fired up the drill, and the superheated, sterilized water began its slow, relentless descent into the ancient ice. For 24 agonizing hours, the drill worked its way down, foot by foot. The team on the surface watched their monitors, tracking the drill head's progress, a tiny point of heat descending into a vast, frozen unknown. They were attempting to enter a world that had been completely isolated from their own for millennia, a world the experts had declared dead. They had bet their careers that it was not. What the camera saw next would rewrite the textbooks on life itself. Life finds a way. On the morning of January 20th, 2013, inside the warmth and controlled chaos of the Whissard Project's mobile command center, the atmosphere was electric with a mixture of exhaustion and anticipation. For a full day and night, the hot water drill had been roaring, consuming thousands of gallons of fuel to send a continuous jet of sterilized water down into the abyss. The team worked in shifts, their faces illuminated by the glow of the screens displaying a constant stream of data: temperature, pressure, descent rate. After years of preparation, they were just a few feet away from breaking through the bottom of the ice sheet. 2,628 feet of solid ice below them lay the dark, mysterious waters of Lake Whillans. The team huddled around the main console, watching the numbers. Then it happened. The monotonous hum of the operation was broken by a shout. The pressure gauge on the drill hose suddenly dropped. The resistance that the ice had been providing vanished. The drill head was hanging in a void. They were in. A wave of cheers and applause erupted through the cramped container, a release of years of pent-up tension and hope. They had breached the ceiling of a lost world. The first phase was complete, but the celebration was short-lived. The most critical part of the mission was just beginning. The clock was ticking. The borehole, a narrow tunnel of water through a mountain of ice, would begin to refreeze the moment they stopped drilling. They had a limited window to deploy their instruments and collect their priceless samples. Immediately, the drill hose was retracted, and a new team sprang into action, preparing the scientific probe for its descent. This sophisticated package of instruments, a gleaming cylinder of titanium and steel, was their messenger to the world below. It was armed with high-definition cameras, powerful LED lights to pierce the eternal darkness, and a suite of sensors known as a CTD for measuring the water's conductivity, temperature, and depth. Most importantly, it carried a series of Niskin bottles, specially designed to open on command at a specific depth, gulp in a sample of the pristine lake water, and then seal themselves tightly for the journey back to the surface. This would protect the samples from any potential contaminants that might be lingering in the borehole itself. As the probe was carefully lowered into the opening, the command center fell silent again. All eyes were on the main video screen. For several minutes, the only view was the shimmering, scalloped walls of the borehole. Then, the camera emerged from the bottom of the ice. The first ever images of the interior of a subglacial lake flickered onto the screen. The view was otherworldly. The camera's powerful lights cut through the pitch-black water, but the beam didn't travel far. It illuminated a murky, tan-colored environment, thick with suspended sediment, swirling like a ghostly fog. This wasn't the crystal-clear, sterile water many had imagined. It looked alive. But stunning visuals were not scientific proof. The real evidence had to come from the water itself. The probe was lowered further into the lake, its sensors collecting data, and at the designated depth, the command was sent. The Niskin bottles snapped shut. The long, slow process of retrieving the probe began. When it finally emerged from the borehole, dripping and cold, it was rushed to the on-site mobile laboratory. A young graduate student named Rachel Morgan was given the honor of performing the initial analysis. She applied a special fluorescent dye that was designed to bind to DNA, causing any living cells to glow brightly under a microscope. She placed the slide onto the microscope stage, adjusted the focus, and leaned in. What she saw made her gasp. She checked the focus again, thinking it must be an error, some dust on the lens. But it was real. The view was not one of a few scattered points of light, but a dense constellation of tiny glowing green dots. It was teeming with life. She called over a senior scientist, who looked into the eyepiece, fell silent for a moment, and then simply said, "Oh, wow." The sterile void theory was not just wrong. It was spectacularly, monumentally wrong. The initial cell counts were staggering. In a field where finding a few hundred cells per milliliter would have been a major discovery, they were finding over 130,000. This wasn't a sparse, barely surviving population on the edge of extinction. It was a density of life comparable to some of the world's deep oceans. In a matter of hours, the Whissard team had gone from hopeful theorists to vindicated pioneers. They had found the first definitive proof of a functioning ecosystem in a subglacial Antarctic lake. The mockery from the old guard, The skepticism from their colleagues had evaporated in the face of this irrefutable, glowing data. Back in their more advanced labs at McMurdo Station, and eventually in the United States, the full scope of the discovery came into focus. Metagenomic sequencing, a powerful technique that analyzes all the genetic material in a sample at once, revealed a stunning diversity of life. They identified nearly 4,000 different species of microorganisms. The dominant group was proteobacteria, but there were also firmicutes and actinobacteria, all ancient lineages of life. These weren't just random microbes, they were specialists, perfectly adapted to their unique, dark, cold environment. The genetic analysis showed they were chemosynthesizers. Instead of using sunlight for energy, they were metabolizing minerals from the fine-grained sediment, eating the iron, sulfur, and ammonium that had been ground up by the glacier, moving over the bedrock for eons. They were the producers at the base of a completely self-contained food web, recycling ancient carbon that had been buried with the lake millions of years ago. They had found an ecosystem running on a completely different engine from the one that powers our world. And that discovery pointed to something far, far bigger than Earth, the alien world within. The discovery of a thriving ecosystem in Lake Whillans was a watershed moment for polar science, a vindication that echoed in labs around the world. But its true significance rippled out far beyond Antarctica, beyond even the confines of terrestrial biology. In the final analysis, the Whillans project had done more than just find microbes in a cold, dark lake. It had provided a powerful and tangible blueprint for how life could exist in the most extreme environments imaginable, both on our planet and, thrillingly, elsewhere in the solar system. For decades, astrobiologists, the scientists who search for life beyond Earth, had been dreaming of places like Jupiter's moon, Europa, and Saturn's moon, Enceladus. Both are tantalizing worlds, covered in a thick shell of water ice. Data from spacecraft like Galileo and Cassini strongly suggest that beneath these frozen shells lie vast liquid water oceans, potentially containing more water than all of Earth's oceans combined. These oceans are not kept liquid by the sun, which is too far away to have much effect, but by the tidal forces of their giant parent planets. The constant stretching and squeezing of the moon's interiors generates enough heat to prevent the water from freezing solid. The question that hung over decades of research, the multi-billion-dollar question, was always the same. Could anything actually live there? Beneath nearly a kilometer of Antarctic ice, a team of scientists drilled into darkness. For years, Lake Whillans had been little more than a radar echo, a ghostly signal detected under the West Antarctic ice sheet. No sunlight had touched its waters for at least 1,000,000 years. To reach it, the researchers of the Whillans project, Whillans Ice Stream Subglacial Access Research Drilling, had to melt their way through layers of sterile ice using a clean, hot water drill designed to prevent even a single surface microbe from contaminating the pristine lake below. They were, in a very real sense, explorers entering a new world, one that hadn't seen open air since before humanity existed. When the drill finally broke through, sensors detected a change in pressure, a pocket of liquid. Then, as the sample tubing drew up its first cloudy droplets, something remarkable happened. The readings spiked. There was oxygen, there were nutrients, and, most shocking of all, there were signs of metabolism. Tiny, living cells. Lake Whillans provided the stunning answer that generations of astrobiologists had dreamed of. Yes, life can thrive in the dark. The microbes found in those subglacial waters weren't relying on sunlight or photosynthesis. Instead, they were part of a vast chemosynthetic ecosystem, one that feeds on chemical reactions between rock and water, much like the microbial colonies living near deep-sea hydrothermal vents. They breathed sulfur and iron instead of oxygen, consumed minerals instead of plants, and turned raw geology into energy. In doing so, they confirmed something profound. Sunlight isn't the only spark that can ignite life. All it takes is liquid water, a source of energy, and time. In the years that followed, those humble microbes changed the direction of space exploration. NASA's Europa Clipper mission, designed to study Jupiter's frozen moon, began rethinking what life might look like beyond Earth. Before Whillans, Scientists thought any chance of extraterrestrial biology depended on sunlight or organic debris from comets. But the Antarctic lake revealed a deeper truth: that entire ecosystems can thrive beneath solid ice, completely sealed from the surface, powered only by chemistry and patience. Europa's vast ocean, buried beneath miles of ice, suddenly looked a lot more alive. Its dark, pressurized waters share nearly identical conditions to those of Lake Whillans. No sunlight, mineral-rich seafloor, and an icy shell above. The Clipper's instruments were redesigned with this new understanding. Instead of searching for pigments or photosynthetic byproducts, they now looked for subtler signs, chemical fingerprints left by chemosynthesis, like unusual ratios of methane, ammonia, or hydrogen sulfide in the moon's vapor plumes. Every sample from Whillans became a map, a living guide for decoding alien chemistry. But the story didn't end there. It circled back to where humanity's curiosity about subglacial life began: Lake Vostok. In the 1990s, when Russian scientists first drilled toward the buried Antarctic giant, they believed they might be the first to reach an untouched body of water sealed for millions of years. What they didn't realize was that their kerosene-fueled drills were contaminating the very environment they sought to study. When the first samples came up containing microbes, Western scientists doubted the results. How could anyone know what was truly native to Vostok and what had leaked in from the surface? Lake Whillans changed that forever. The American success with sterile, contamination-free drilling gave researchers a new lens to revisit old data. In a rare act of collaboration between former Cold War rivals, U.S. and Russian teams re-examined the Vostok samples using modern genetic sequencing and the contamination controls perfected in the Whillans project. What they found was astonishing. Even after decades of skepticism, many of the microbes in the Vostok ice cores were indeed unique, native life forms that had evolved in total isolation beneath Antarctica's ice. The Soviets, it turned out, had been right all along. Whillans had become the Rosetta Stone, the key that unlocked the truth hidden in Vostok's frozen depths. For most people, it's easy to think that discoveries like this happen in a single cinematic moment: a test tube under a microscope, a flash of realization, a headline that changes everything. But in reality, the discovery of life beneath the ice was the slow burn of decades of perseverance, failure, and obsession. Scientists debated for years whether life could even exist under such crushing pressure, cut off from sunlight for millions of years. Each failed expedition, each contaminated sample, was another chapter in a long, stubborn story of curiosity refusing to die. And maybe that's fitting, because the life inside Lake Whillans mirrors the scientists who discovered it, both surviving in darkness, both proving that persistence can overcome impossible odds. The organisms themselves are something out of time. Genetic analysis suggests that some of them may have been evolving in isolation for 15 million years. That's before humans, before mammoths, even before the ice sheets themselves locked Antarctica in their frozen grip. These microbes are evolutionary orphans, branches of the tree of life that grew in silence, far from sunlight, under crushing weight and eternal night. They are, quite literally, snapshots of an Earth that no longer exists. Through them, scientists can glimpse how life might have begun, not just on this planet, but anywhere. Maybe the same process happened on Europa, or on Saturn's moon Enceladus, or in some distant icy world orbiting another star. Maybe the pattern repeats: rock meets water, water meets time. And from the chemical hum of existence, life emerges. The most powerful lesson of Lake Whillans isn't just that life can exist in the dark. It's that life finds a way to persist where logic says it shouldn't. From the silent depths of Antarctica to the frozen moons of Jupiter, the discovery built a bridge between worlds. One that connects our fragile planet to the vast, unseen ecosystems that may lie waiting across the cosmos. The once impossible dream of finding alien life suddenly felt closer, almost within reach. And yet, perhaps the most humbling revelation is this: the first true analog for alien life was never light years away. It was here all along, hidden beneath our own feet, whispering its quiet proof through the ice. In a world obsessed with exploring other planets, Lake Whillans reminded us of something profound. Before we can understand life beyond Earth, we have to look deeper into our own. Because sometimes the universe doesn't send answers from the stars, it hides them in the coldest, darkest corners of home. Did the persistence of US scientists just hand us the roadmap to finding alien life? Or did we just awaken a world that was meant to stay hidden? Let us know your thoughts in the comments below. And if you want to explore more incredible scientific frontiers, be sure to like this video and subscribe.